Hello, everyone. You're welcome back to the channel. Compliments of this great season. It's wonderful to have you around. This morning, we are discussing something very quickly. We want to discuss genetics in bacteria or gene transfer in bacteria. And this is an important concept. It's quite easy to understand, but some do find it difficult. Now, bacteria, there are two types of transfer of genetic materials. There's what you call a vertical transfer and a horizontal gene transfer. Now, vertical is up to down, horizontal is sideways. Now, when you talk about a vertical gene transfer in bacteria, you're just talking about the transfer of genetic materials, the genetic constitution, genetic composition from the mother to her two daughter cells. So, many times bacteria are engaged in binary fission. So one cell divides to become two daughter cells. One mother cell divides to become two daughter cells. And in this division, what you have happening is that the total genetic component in the mother cell is replicated into the two daughter cells. Simple, easy peasy. And that's what you call the vertical genetic transfer. So transfer of genes from mother to two daughter cells, simple. Now, secondly, there's also what we call horizontal gene transfer. So when you think of horizontal, you think of it as side to side. So that's the transfer of genetic material from one bacteria to another. Now, not from mother to daughters, but from one bacteria to another bacteria. And there are about three methods by which this thing happens, as this transfer of genetic materials happens. There is transformation, there's transduction, and there's conjugation. These are the three methods. Now, they are easy to understand and you must understand them. When it comes to transformation, transformation is the transfer of genetic materials from one bacteria to the other. But what happens in DNA transformation is that one of the bacteria is alive while the second one may have died, but the idea is that its DNA is in the environment. So what you have actually happening is that the bacteria that is living takes up DNA of the second bacteria, but that second bacteria may be dead. So its DNA is in an environment. It could be in a solution of water, it could be anywhere, but it's in the environment. And then the living bacteria takes up the DNA of the dead bacteria and it takes it up through its pores, through its membrane, and then incorporates that DNA into itself. That's what we call DNA transformation. I'll do a video where I'll talk about the Griffiths experiment. This is the experiment that was used to explain how this occurs in nature. But it's a simple process, DNA transformation. What you have is a live organism, a bacteria, takes up DNA of another bacteria, but that other bacteria is dead and its DNA is in a solution. But the living bacteria is able to take up that DNA through its pores, through its membranes, and then incorporate that DNA into its own DNA to form a new DNA. And then it becomes a transformed organism. And so that's why we call it DNA transformation. This happens a lot in nature. Now, secondly, you have conjugation. In conjugation, I used to try to tell my students that think about it like as DNA having sexual intercourse. Two DNA stay side by, I said two DNA, two organisms, they stay side by side. And one of them is able to give its part of its genetic materials to the second one. And this is mediated through what you call a sex pilus, which is like a passage. So the donor organism creates a passage because it has what you call F plasmid. That is a type of plasmid. Plasmids are what we call extra chromosomal DNAs. They are DNAs that are not part of the chromosomal DNA in a bacteria. So one of the bacteria then, because it has F plasmids, which we call fertility plasmids, is able to produce a sex pilus, which is a passage between itself and another bacteria. Now, when it produces this sex pilus, through this passage is able to send genetic material to the recipient. So it's a very easy process. So think about it as two bacteria coming together and exchanging genetic materials. Remember that in transformation, one of the bacteria, the DNA is in solution in the environment. The living one takes up that DNA. In this second one, the two bacteria, they are living, but one is a donor, the second is a recipient, and the donor has fertility plasmid, F plasmid, and he uses it to create a sex pilus, and it attaches itself to the recipient and gives it the DNA. Simple, it's not difficult. And the third one is transduction. And in transduction, what happens is that there's a transfer of genetic materials from bacteria A to bacteria B through a virus, what we call a bacteriophage, which are viruses. So the virus is able to be in this DNA, infect this DNA, invade this DNA, take part of its genetic material, and then go and infect another bacteria. So take DNA from bacteria B and carry it to bacteria A, or from A to B, anyway, anyhow. So that's what you need to understand, that you can have transfer of genetic materials from one bacteria to another using a virus, what we refer to as a bacteriophage, which is a virus that is able to invade a bacteria. That's what you have happening in transduction. We'll still discuss types of transductions in depth. There's generalized transduction, there's specific transduction. There are different types of transduction. You also have conjugation, then you have transformation. These three methods are methods for horizontal transfer of DNA. That's DNA from organism A to organism B. Simple and easy peasy. Thank you for listening, and do have a beautiful day. Remember to like, subscribe to our channel. Have a beautiful day.